Hello, my name is Saloni. Hopefully you already know that. And my presentation today is called How Many Times Can We Roll Nature's Dice? If all goes well, there should be a screen over here that has my slides, and hopefully everyone can follow along. So genetic engineering and genetic editing are powerful techniques that have been used to understand biological systems. The idea of editing a person's genome has been around in science fiction since the late 1800s, but it took another 100 years for real science to make a breakthrough in this area. I wanted to talk today about the steps that we've made since then and where we can go from here. Each child born today is a unique, untested genetic experiment. Mother Nature throws up a combination of genes and a human is pieced together from there. But science has decided that it wants some control in this process. We have only just begun to scratch the surface of what is possible, and decisions that we make today can influence the course of the world permanently. So first, a little bit of background. Restriction endonucleases were discovered in 1970. These enzymes are natively found in cells, and they defend bacterial cells against invading pathogens by binding to specific sequences and then creating a double-stranded break in the foreign DNA. The idea of intentionally breaking DNA was something that human researchers were very interested in replicating, and 25 years later, researchers at Johns Hopkins University managed to do exactly that. They developed zinc finger nucleases, which are restriction enzymes that are engineered to recognize specific DNA sequences and are able to cleave these sequences with high specificity. Zinc finger nucleases were used for editing over the next couple of decades, but side by side, there was increased understanding of a molecule called CRISPR, which stands for Clustered Regularly Interspersed Short Palindromic Repeats. In 2012, a molecule called Cas9 was discovered, and putting these two together led to an explosion of interest in the human genome editing process. So I'm going to show you a short video depicting the CRISPR-Cas9 process. And so the Cas9 complex is shown here in green, and then the CRISPR sequence is transcribed into an RNA or crRNA in purple. And so the crRNA will locate its complementary sequence on the target DNA, and then Cas9 will bind to short sequences on the target DNA called PAM, and then cleave the DNA. Cleavage will cause double-stranded breaks, and at this break site, DNA can be added, removed, or modified, and these mutations are both efficient and specific. Because CRISPR is also a relatively simple tool to generate, this is the best available tool that we currently have on the market. Since its discovery, CRISPR has been used in thousands of plant and animal studies to understand the relationship between genes and disease, and clinical trials are also beginning in humans to show the safety of CRISPR. More importantly, with this key discovery, the idea of enhancing the natural system, or creating something within the DNA from scratch, was brought to the forefront, and human germline modification became a reality. In November 2018, a Chinese researcher, He Jiankyu, shocked the world when he announced that he had created the world's first genetically modified humans. It came to light that he had injected CRISPR molecules into 31 embryos with the goal of deleting the CCR5 protein. This protein is located on the surface of white blood cells and can act as a portal for HIV molecules to enter the cell. And so deletion of the CCR5 proteins could potentially prevent a person from contracting HIV. So his clinical trial resulted in the birth of two twins, Lulu and Nana, but unfortunately he didn't receive the reception from the scientific community that he expected. Instead, the international community was shocked and horrified, and they condemned his research. Because HIV and AIDS are preventable and they are treatable to the point where viral loads are undetectable, researchers around the world believe that this research didn't meet the threshold of serious unmet medical need that would actually justify embryonic editing. In this case, the precautionary principle, which states that action shouldn't be taken until all consequences are known, would thoroughly resist this kind of work, given that not enough preliminary research was done beforehand. Leaders in the field of genome editing collaboratively published a 2015 paper in Science, stating clearly that their recommendation would be to discourage any attempts at germline genome modification until societal, environmental, and ethical implications of such activities are discussed among scientists and governmental organizations. However, following the birth of the twins in 2018, one of the original developers of CRISPR believes that the genie is out of the bottle and it is too late to stop all research like this. The problem is that by the researchers' own admission, the twin girls born are genetic mosaics, meaning that some cells do contain successful deletion of the CCR5 protein, but some cells do not. So the girls are actually still susceptible to contracting HIV, and the experiment was not quite a success. So how much research are you actually allowed to do in this area? Human germline editing is completely banned in Canada and the US. Even research that involves editing of embryos less than 2 weeks old, without any intent for implantation, is tightly controlled, as is the case in parts of Europe. Although the rules are less stringent in China, He Jiankyu was sentenced to 3 years of jail time for the work that he had carried out. However, court documents have made note of the birth of a third genetically edited baby, something that Jiankyu had previously mentioned but not confirmed. For better or for worse, we now have 3 little byproducts of human curiosity crawling around our planet. So we know that the technique is there to perform human germline editing, but these techniques cannot be performed in a vacuum. We need to ask ourselves, although genetic editing can be performed, should it? A growing number of people say yes, that having a baby, your own little copy, is too important to leave up to nature or to God or to random chance. But before we open the floodgates, there are 3 important questions that I wanted to discuss. Firstly, what exactly is a disease or a defect, and who gets to decide whether something needs to be treated? For example, within the deaf community, it is a long-standing point of frustration that the public perception is that deafness is something that needs to be treated. However, individuals who are deaf believe that sign language is a cultural cornerstone, and to encourage a deaf child to learn how to lip read or to get cochlear implants would be considered erasure of that culture. Affected individuals themselves in this case would resist genetic correction. Secondly, can diseases contribute to an individual's well-being? Bipolar disorder has high genetic association and is characterized by alternating periods of mania and depression. During periods of mania, sufferers often experience periods of significant energy and creativity, but during periods of depression, sufferers are prone to self-harm and suicide. Celebrities like Catherine Zeta-Jones, Mariah Carey, and Kanye West have attributed some of their success to their bipolar disorder because of the periods of mania. However, bipolar disorder is a heterogeneous experience, and it is impossible to predict how each individual will be affected when you're just looking at the embryos. In addition, certain gene variants that predispose us to specific conditions may actually protect us from other conditions. Fitness advantages and disadvantages often have a way of balancing each other out. The major histocompatibility complex, which is responsible for many immune responses, is filled with mutations, but these mutations actually allow for other diseases to emerge in the population at lower rates than the frequency of their deleterious alleles might suggest. It looks like there's a joint relationship between the mutations and the lower frequency of disease, and so removing the mutations might actually increase the prevalence of diseases like cancer and schizophrenia. Thirdly, how should we delineate the boundary between enhancement and treatment? Already these lines are thin. Vaccinations, for example, don't treat an illness, but they enhance the body in order to prevent future illness. Another example would be that in a 2004 study, it was found that for every 1-inch increase in height, salary increased by approximately $1,000. Although this is not a linear correlation, it does appear that there is some relationship between a seemingly cosmetic factor like height and a tangible lifestyle outcome like salary. The last important consideration is one of safety. Already, the first human genome experiments didn't achieve the results that the researcher was aiming for, and this could happen again in the future. This is quite a broad topic, but quickly, 4 main points of consideration. 1, it is possible that an alteration of one small gene in the body could lead to unintended effects on the rest of the system. Secondly, the individuals that undergo genetic editing, the embryos that actually become real people eventually, aren't the ones that provide consent to their own editing. This is done by their parents, even though it is the embryos that must bear the risks and the consequences of editing. Furthermore, the choices of modifications could be nearly limitless. Parents might be able to choose their child's height, their child's favorite color, or even their child's sexuality, certainly a problematic endeavor. Lastly, it is very difficult to perform rigorous testing. Both animal and in vitro cell models don't approximate human physiological conditions closely enough to be able to draw concrete conclusions, and it would be unethical to perform human testing without these preliminary measures. So far, we've been discussing the advantages and limitations of gene therapy as it applies to a single individual. However, it is also possible that genetic editing could be used to benefit society as a whole. About 40 genes have been associated with anger and aggression, the most robust association being with the monoamine oxidase A gene. A Finnish study, which examined 2 independent cohorts of prisoners, found that a low-activity genotype of this gene was highly associated with extremely violent behavior. Once causation is better established, it does seem to make sense to look into research that would remove violent offenders from society by simply removing the violent personality itself. Interestingly, or maybe concerningly, a study performed at Illinois State University found that at least 40% of individuals surveyed supported policies that would restrict reproductive access for people who are poor, unintelligent, or criminally inclined, and these are quite radical conclusions to draw. So we've talked about safety, and we had a little bit of a should we, shouldn't we discussion about whether or not a genetic editing can be performed. But let's now talk about where genetic editing can realistically go from here and how we can move forward. Although it is currently banned, if it were made legal tomorrow, what would be the first steps that we would take? And for the answer to that, we look at monogenic diseases. These are diseases that are caused by only one specific mutation within one specific gene. For examples, Huntington's disease, Tay-Sachs disease, cystic fibrosis, and sickle cell anemia. None of these diseases have cures, and they all lead to shortened lifespans. Any medical intervention is focused only on managing symptoms rather than actually treating the disease. It seems logical then to involve CRISPR in finding solutions for these types of monogenic diseases. We know it is possible, we just need the political will to get us there. It is not hard to imagine that a world without these diseases is just around the corner, and editing genes associated with more complex diseases will follow shortly after. I hope it's been made clear that alongside the development of scientific techniques, it is equally important to have conversations about the ethical implications and the future possibilities. Although it is far too likely that uncontrolled access to this technology would lead to destruction of the human race, I wanted to walk you through one possible, slightly more optimistic scenario of the future. Welcome to 2220. In this strange new world, humans have become no less susceptible to Moore's Law than a computer from 2020. Humans from 2020 would probably have a hard time differentiating between a computer from 2220 and a 2220 human. Progress began slowly. Pressures by billionaires like Elon Musk and Jeff Bezos, who were always incentivized by their wallets, led to the US government repealing their ban on embryonic editing, and other nations soon followed, not wanting to be left behind. Every scientist started experimenting with CRISPR, and progress occurred exponentially. They mapped out associations for all known genetic diseases, which let them manipulate embryos to correct any defects or germline mutations. Expansion of right to try legislature provided first access to couples with highly heritable genetic diseases, but pretty quickly, any couple who could afford it started doing IVF and pre-implantation screening. The idea of loading the genetic dice instead of just rolling it became a prominent point of discussion, but some people were still wary. Pre-implantation screening seemed like a good middle ground. It wasn't engineering, it was sorting through all of the garbage that Mother Nature had thrown up and then choosing the best combination. After all, no one wants to start their baby off on the wrong foot. Progress was also being made in synthetic embryonic engineering. Scientists began experimenting with the bare minimum number of genes needed to create a functioning organism. Each gene within a genome was deleted separately so that DNA, filled with redundancies and duplications, could be streamlined. The first genome to be entirely dissected and put back together on a synthetic chromosome was a small bacterial cell, and amazingly, it was capable of self-replication. Then, the first eukaryote, a yeast cell, was minimized. Complex eukaryotes, like dogs, were further down the line, but eventually their 3 billion base pairs were reduced by half and then by half again. Unfortunately, these beings ended up being genetic dead ends. They were incapable of reproduction or even normal lifespans, and no solution could ever be found. Minimal genome construction was instead used to further the development of naturally created humans. Scientists figured out how to insert entirely new genetic information onto a scaffold and construct new genes out of something that wasn't there previously. For the first time, the entire human biosphere was programmable. Experimenting with spare embryonic tissue, originally intended for implantation, was permitted. Traits like height, metabolism, and intelligence were refined. Slowly, emotions were also altered. Negative emotions like anger were dispensed with and replaced with the rainbow of bliss. Although some people protested, the overwhelming majority were happy for their progeny to be able to opt in to a more meaningful existence. Genetic improvements were all well and good, but the thirst for extraordinary skill and abilities left the human race chasing after more. Regulatory red tape on the medical side meant that technology had advanced much further than biology, and now the task became to merge the two. Humans began to bootstrap themselves to a new superintelligence. Prosthetic limbs and other engineered parts were far superior to biological ones. Quite soon, individuals began to request voluntary limb replacements and other organs so that they could also benefit from things like superspeed and superintelligence. People were limited only by the extent of their own creativity and wallet. There was a brief moment of unprecedented prosperity before reality hit. It turns out that whatever genetic editing had been performed, in conjunction with all of the organ replacements, had permanently messed with something important in germline cells. It took a few more generations for the problems to become evident, and by then it was too late to go back and start over. Something about these edited individuals, these first-class, these top-flight individuals, didn't quite lead to high reproductive success, and so every few generations, the second class of humans, C2, came in handy. C2 originally started out as a group of humans who opposed the biohacking movement, couldn't afford access, or simply jumped on board too late. Now, their descendants are the only thing keeping the human race afloat. Mating between the first class and the C2 individuals in alternate generations ensures that the first class is able to keep going. 500,000 years of evolution, and it turns out that we should have stopped a little after 2020. Can you believe that? Humans were successful in transcending their own prior existence on Earth, but not their own genetics. Forward thinkers of the first class are hoping to start a breeding colony of C2s in hopes that a second go-around will lead to more success. Here's to hoping for better dice to toss. To close out this presentation, I have my references, and then I also just wanted to say thank you to Shauna Pandya, Gary Goldsand, and David Pierce. Their lectures inspired a lot of the content in this presentation. Thank you also to Dr. Soles from encouraging me to stay away from Team Doom and Gloom, and then for his mentorship throughout this semester.